Hello, in this video we're going to take a look at the anatomy and function of the male and female reproductive structure, including accessory structures. We are in chapter 27, objective 2. So we're first going to take a look at the male, and then at that point I'm going to create another video that's just over the female. So we'll say 2A is male, 2B will be the female reproductive system. So let's go ahead and get started on the male reproductive system. You should recognize the structures from lab. So I'm not going to go over the structures with y'all. You should just know the structures. You're going to have to definitely know them for the lab class. I encourage you all to watch this video. You've got a handout in your packet that you got from the bookstore that goes over the male reproductive structures, just an overview of that. It's from Armando Hasuda again. I encourage y'all to take a look at that, especially for lab class as well. Now, the function of the male reproductive system, you're definitely going to have to know this. It's going to be very similar to the female, a few little differences there. It does produce gametes, which again, for male, are going to be the sperm cells, right? Stores gametes, again, it stores sperm cells, nourishes gametes. So when we're talking about the male, we're talking about the sperm cells, and then transports those to the female. So I do want to show you something. It's just kind of interesting. This is fairly new, it was 2020. They discovered that sperm actually roll rather than swim. The music is a little intense, so I don't know if you're going to be able to hear the music, and that's fine. You can listen to it on your own, but there's no talking. It's just music on this. But to show you the movement that sperm takes as it moves is pretty amazing. So again, it's more of a rolling action than a swimming action. So I will let you kind of take a look at the rest of that on your own. I just wanted to kind of show you where the new information has come from. Whoop, that's still pretty loud on the music. Again, the music is interesting, but anyway, pretty cool little video to explain the movement of sperm. The functions of major structures. So again, if you're not sure where these structures are, go back to that first slide where all the structures are listed. You do need to know the functions of these specific structures. So the testes, that is the site of sperm production. The scrotum or the scrotal sac, that is the skin that surrounds and protects the testes. The epididymis, this is the part that is the storage site for sperm. Vas deferens, a passageway for sperm during ejaculation that carries the sperm from the epididymis to the urethra. The urethra is also a passageway for urine, so that will carry both reproductive and urinary fluids. Seminal vesicles produce semen, which is the fluid, the nourishing fluid that's going to mix with sperm. Now, there are other fluids that are provided for the sperm. Seminal vesicles is just one of those structures. So again, take a look at that. Make sure you know that from lab. The next thing I want to do is look at this anatomy of the testes. So I know, sorry about my horrible screen page here. It's just like a hot mess there. So let's go ahead and in your packet, I'm going to turn that just a little bit so we can see. In your packet, you have got this handout here, and I did want to kind of go through that with you. So I'm on page, it's around 231, and just to give you an idea where we are, but to kind of take a look at some of this, you've got the testis anatomy here. Making sure you can see what we're doing here. Now, within the testes, you've got these structures called seminiferous tubules. So as you can see, they are tubular structures here, and these are found within the testes, and the function of the seminiferous tubules is actually where sperm are made. Now, they're created, I'm going to put a little 1 right here, because they're created in these seminiferous tubules, And then they're going to move to what's called the rete testes. So this area right here, I'll get a different color so you can kind of see. I don't know if you can see the different color or not. But this right here would be the rete testes. And it's going to be right here. So we're going to call that number 2. So again, the sperm are going to go from, they're going to be created in the seminiferous tubules. They are then going to be moved to the rete testes. And then from there, they're going to move into the epididymis. Now, the epididymis is going to be divided into 3 sections. Not going to worry too much about it, but you can see these little passageways here, which are going to carry that sperm into the epididymis. So you've got your head of the epididymis, body of the epididymis, tail of the epididymis. So they're going to start in the head of the epididymis. They're going to arrive there first, so we'll call this 3A. Then they're going to go into the body, which would be 3B, and then they're going to end up in the tail. So they're going to be in the tail last, and the tail is going to connect to, you can't really see in this one because it goes behind, so it's going to go behind all of this right here, and that's where that's going to end up going. That is the vas deferens. So this is going to connect to the vas deferens. We'll call that number 4. And the vas deferens then is going to go up inside the body. So this is going to be a passageway that is going to be carrying mature sperm to the urethra. That's where it's headed. So let's take a look at, if you can see that a little bit. Let me switch that up. Let's take a look at a cross section of the seminiferous tubule and kind of see what's going on in this picture here. So you've got your seminiferous tubule, which is this structure right here. So you kind of have a cross section of it, and that's what you're looking at. There's the lumen on the inside. So this is where those mature sperm are going to enter. So you're going to kind of start at the outside as far as the sperm maturation goes and work your way in. So they're going to start on the outside layer as immature, and as you move towards the center, they're going to become more mature. Now I want to point out 2 cell types here because these are important. You've got your Leydig cells right here, also known as interstitial cells. And the job of these interstitial cells or Leydig cells is to produce testosterone. Now the other cell type that you're going to see that we're going to talk about are the Sertoli cells. So this whole thing right here, this is a Sertoli cell. Sertoli cells, also known as nurse cells. These are located within the tubule. Notice the Leydig cells are located outside in the interstitial space, right? They're not within the seminiferous tubule. They're on the outside of the seminiferous tubule. These Sertoli cells are located within the tubule. I'm just going to put tubule there. Hopefully you know that means seminiferous tubule. And their job is to help with the synthesis of sperm. So you've got to have, we'll talk more about this in a minute, but you've got to have the testosterone, which is the hormone that is going to help with sperm production. And the Sertoli cells are going to help with sperm production. These spermatogonium right here, this is a cell type. Notice it doesn't have a tail, it's just a cell right here. And then as you move into the lumen, you can see that tail on that sperm start to form. So on your next page, let's take a look at this page. This is your male sperm production. So you've got 2 main processes involved in sperm production. You have spermatocytogenesis, and then you have spermiogenesis. So those are your 2 main two main processes involved. Now let's kind of take a look at spermatocytogenesis first. And where are we as far as in the seminiferous tubule and that kind of stuff. So this is the outermost layer of the seminiferous tubule. Where am I going to write that? Here we go. And as you move down, you're going to be in the lumen. So again, you're going to work your way from outside to inside, okay? Where again, you're going to have the most mature sperm in the lumen part of the seminiferous tubule. So you're going to have your spermatogonia cell that is going to turn into a spermatocyte, and you're going to have primary and secondary spermatocytes. You're going to have spermatocyte 1 and spermatocyte 2. So these are your primary spermatocytes and your secondary spermatocytes. This is all part of spermatocytogenesis, and then that will then turn into a spermatid. Now your spermatids are the structure that's going to begin spermiogenesis. Spermatids are immature, where the spermatozoa are going to be mature, also known as sperm. So you definitely need to know your order of cells that are going to take place and what stage of sperm production they're going to occur in. I'm not going to worry about the days, okay, as far as how long it takes for these things to occur. I'm not going to worry too much about that part as far as testing purposes. So let's take a look at your next page here. Hopefully that printed out okay and you guys can see that. So spermatogenesis, also known as the synthesis of sperm, again, is divided up into two phases. Now, as far as the spermatogonium cells, so if you look at this picture down here, this is the lumen, okay, so these are going to be the most mature, and your spermatogonium, or spermatogonia, as it was stated on the previous page, we had A and B. These are going to be your, basically, stem cells. This is going to represent the outermost layer of the tubule, and that's going to work its way inward to the lumen. So it's kind of backwards on this page, but you kind of have to get your bearings and see where you are. So again, these spermatogonia, these sperm stem cells, are going to then mature as they move inward. You've got this Sertoli cell here, also known as that nurse cell. Remember, it was on the inside of the seminiferous tubule. That spermatogonia cell, that stem cell, is then going to differentiate into your primary spermatocyte and your secondary spermatocyte. And at that point, when the secondary spermatocyte, you're going to get these spermatids, and they're going to then differentiate into these spermatozoa, and that's going to be that mature sperm. Those interstitial cells, if you remember from before, those interstitial cells are also called Leydig cells. Yeah, y'all can still see L-A-Y-D-I-G. Leydig cells. And notice they are sitting outside of that seminiferous tubule. There's the lumen, okay, and those interstitial cells are on the outside of the seminiferous tubule, with the Sertoli cells being on the inside of the seminiferous tubule. And then we'll come back to this when we get to female reproduction. I don't know why it does that, but okay. There we go. So, spermatocytogenesis, which we've had the previous picture that we discussed, spermatocytogenesis, like we said, was divided into, or spermatogenesis was divided into two phases. Spermatocytogenesis, which was the first phase, and this is where we're going to get those spermatogonium, A and B, your primary spermatogonium and your secondary spermatogonium, those stem cells, and they're going to form spermatocytes, which are then going to form spermatids, which are then going to eventually form sperm, also known as spermatozoa. So what's happening here? You've got your spermatogonium stem cells here that are going to undergo mitosis. That's what that 2N right there means. It means it's diploid. It's got basically twice the number of chromosomes within that cell. Okay, it's got the set from mom and the set from dad. That's going to undergo mitosis, and you're going to end up with two genetically identical cells. That's what mitosis does. Now, those primary spermatogonium, they're going to undergo division, but notice this time they're going to undergo what's called meiosis. And look what happens during meiosis when you go from the primary to the secondary here. You're going to go from a diploid cell to a haploid cell. This means you're going to have half the number of chromosomes. These are not genetically identical. So during this process of the spermatogonium, you are undergoing meiosis, in which you're ending up with two cells for meiosis 1 that have half the number of chromosomes. And the end result from meiosis 1 and meiosis 2, the end result of meiosis is you're going to have these immature spermatids. Notice they don't have tails. You're going to have 4 genetically different cells. And then those 4 genetically different cells are going to mature into 4 genetically different haploid sperm cells. Okay? So this is just the spermatid to the sperm or spermatozoa. Remember, this was spermiogenesis is what's happening there. So that was all that picture was showing. So again, you need to know the order that these sperm go. We talked about that previously. The spermatozoa right here are going to enter into the lumen of the seminiferous tubule. They're going to then, this tubule right here, they're going to enter the rete testi, which is right here. You're going to get these structures that are going to allow those sperm to exit the testi and go into the epididymis. So you're not seeing the epididymis here, but it's first going to go to the head of the epididymis, then to the body of the epididymis, then to the tail of the epididymis, which is then going to connect to the vas deferens, which is a tube that's going to carry the sperm through the body until it reaches the urethra. So again, the accessory glands of the male reproductive system. We mentioned previously that, yeah, the seminal vesicles, you're going to have a right and a left one. They're going to secrete seminal fluid, which is going to be added to the sperm arriving from the vas deferens. So the vas deferens is kind of emptying into those, or the seminal vesicles are kind of dumping into that vas deferens and supplying fluid there. And this is going to make up 60% of the sperm volume, semen volume. So at this point, you're going to have a lot of fluid, which is going to provide energy and movement to the sperm. Now, you've also got the prostate gland. So after the seminal vesicle is going to add the fluid, the next place you're going to get more fluid is going to be from the prostate gland. It's going to pass into the ejaculatory duct, which is going to pass through the prostate gland. So if you remember looking at that on your lab, the prostate gland produces this stuff called prostate fluid. I'm not going to ask you specifically what's in it, but understand that it does contribute to volume, and it is muscular, so it's going to provide that force as well, the prostate gland, to help propel the sperm into the urethra and through the urethra. Remember, the male urethra is longer than the female urethra. Now, you're also going to have fluid coming from the bulbourethral gland. This is an alkaline mucus type substance. It's going to help neutralize acids that could be present in the urethra from the urine residue. Now, remember, in males, the urethra is also for urine transport as well. So to help neutralize that, that's what the mucus from the bulbourethral gland will help with. It also helps lubricate the urethra. So again, you're going to have fluids coming from these three structures within the male reproductive system. So you do need to know what the structures are, overall what they do, and volume, okay? So this right here was just an overview. Again, you should remember that from your lab class. So that's going to conclude the male part, and then there will be another separate video for the female reproductive system.